Hello, this is Bipana Tamang. The topic of my research is evaluating the impact of soil microplastic in broccoli seedling growth, biomass accumulation, and its hormone production. Microplastics are plastic particles that are less than 5 mm in size, which have become a growing concern in terrestrial ecosystem, particularly in the agricultural soil. These particles persist in environment and can alter soil structure and nutrient dynamics. Previous study has suggested that microplastic affect plant growth by disrupting soil water retention, nutrient availability, and hormonal balance. Commercial microplastic with uniform shape and size tend to behave differently in soil compared to self-powdered microplastics, which are irregular and more representative of real world pollution. This study hypothesizes that both commercial and self-powdered microplastics will negatively impact broccoli seeding growth, IA production, and its biomass accumulation, with commercial microplastics showing more pronounced effect. For methods, polystyrene beads were grounded into fine particles to create self-powdered microplastics and weighed alongside with commercially available DFPE polystyrene microplastics. Four treatment groups were established: control with no microplastics, 0.1% commercial microplastics, and 0.5% and 0.1% self-powdered microplastics, with each group containing 40 biological replicates. The microplastics were thoroughly mixed in peat pellets and the trays were labeled accordingly. Broccoli seeds were surface sterilized and sown in pallets, 4 seeds per pallet. The seeded trays were kept in the dark for 3 days to promote germination, then exposed to light and irrigated every 4 days with tap water. Seedling height was measured weekly from 27 to 32 biological replicates per group, with final measurements recorded at 14 days. After harvesting, fresh biomass was measured immediately and samples were dried at 60 degrees for 48 hours to determine dry biomass. For IA quantification, oven-dried samples were rehydrated, homogenized, and analyzed using Salowski assay with absorbance measured at 535 nanometer. Soil pH was measured at the beginning and end of the experiment using pH indicator strips. For results, broccoli seedlings in the control group consistently showed the greatest height in both weeks, followed by 0.1% self-powdered and 0.1% commercial microplastic treatments. The 0.5% self-powder group had significantly shorter plants. The same results was seen in biomass accumulation and IA production. A gradual decrease in plant biomass and IA production was observed across treatments, with the control group exhibiting the highest biomass and highest IA production, whereas the 0.5% self-powdered microplastic treatment resulting in the lowest biomass and lowest IA production. No change in soil pH was observed. In conclusion, this study concludes that both commercial and self-powdered microplastics negatively impact plant health, with 0.5% self-powdered microplastics showing more pronounced effect. Minimal difference was seen between 0.1% commercial microplastics and 0.1% self-powdered microplastic. Nonetheless, all the microplastic treatment group decreased the seedling's height, biomass accumulation, and the hormone production (IAA). For future directions, investigate the long-term effect of microplastics exposure on plant growth and soil chemistry to capture cumulative impacts. Incorporate various plastic types, shapes, size, and include a wider range of microplastic concentration to identify threshold levels that significantly impact plant health. Also examine how microplastic disrupt hormonal pathway, particularly IAA synthesis. In the end, I would like to thank Jessie, Marla, Crystal, and Mike for guiding and supporting throughout this whole research. I would also like to thank my classmates who helped me and support me during this all these weeks. Thank you.